Good evening and welcome to SciCam, the International Foundation for the Protection of the European Dark Bee. My name is Andrew Brown, board member of SciCam. I'm managing director of the B4 project, which is a community interest company based in Cornwall in the southwest of England. B4, Bringing Back Black Bees, represents a group of beekeepers whose aim is to protect the UK's native dark honeybee, Apis mellifera mellifera. Over the past 4 years, the B4 project has been partner in a University of Plymouth Natural Environment Research Council's PhD, investigating the genomic and phenotypic basis of local adaptation in the black honeybee. I have been the non-academic supervisor. It has been a privilege for B4 to contribute, albeit in a minor way, to the work of Victoria Buswell, Professor Mari Knight, and Dr. John Ellis. Dr. Knight is a professor in evolutionary ecology, University of Plymouth. And for most of the time I have known Mari, she has been head of the School of Biological and Marine Science at the University of Bristol. Quoting from Mari's research interests, my research spans a broad range of specific questions from reproductive isolation and gene flow between populations to ecological issues such as dispersal and foraging ranges through to species delineation and phylogeny reconstruction. I am increasingly interested in how these processes operate in populations that are declining and thus of conservation concern, and how molecular techniques can inform conservation science. We are very pleased to have Mari here as host for Victoria Buswell's presentation. Victoria Buswell's presentation is UK AMM populations and introgression. So it's my pleasure to hand over to Mari.
Thank you very much, Andrew. I was slightly worried about what I might have said on my website, so I'm glad it wasn't anything too incriminating. So thank you very much, and it's a real pleasure for us all to be here, but particularly for us to be able to introduce Vic and her project. I'd like to first very briefly also introduce my long-term collaborator here, Dr. John Ellis. So John's worked for us, I'm sure some of you already know, has worked on bumblebee systems for a very long time. I think John probably for over 15 years, I was trying to work out. Also has an interest in knowledge in a broad range of other systems, but we've been collaborating particularly over a very long time period on bee systems, and actually this is our first project looking specifically at honeybee systems. And John obviously sits on the scientific board of SciCam, so he's a good person for everyone to know, obviously. So I don't want to go on too long with introductions because Vic's obviously got an interesting talk to give, but it's my great pleasure to introduce Vic Buswell, who arrived in Plymouth to work with us in 2017, so has been working on this project now for several years and is just getting to the really exciting bit where she's got some results to show. Vic came to us from the University of Canterbury, where she was studying for a degree in biosciences, where she'd done some interesting work on, as part of that degree, had done some interesting work on earwigs, I understand, and the molecular ecology of those in orchard systems. But has a very broad range in entomology, so she, as well as a background in molecular ecology, so she was really well suited for this particular project. And it's been a real pleasure working with Vic over the last few years. And so without further ado, Vic, please take the floor and tell us what you've been doing over that time.
Thanks very much. I'm just going to share my screen. Just let me know if you can see the right part, Mari.
Yeah.
What can you see there?
So I think you need to switch. Yeah, perfect. Yep.
There we go.
No, I think you've switched back again. So
switched the wrong way. God.
There. Yeah.
Yeah. Are we good?
Okay, we're good. Thanks, Vic.
Perfect. I'll just put this down to speaker view so I can tuck you into a corner somewhere. Okay, so yeah, as mentioned, my name's Victoria, and we are actually, all of us are based at the University of Plymouth. So I'm going to be speaking about British populations of European dark bee, or Apis mellifera mellifera, and introgression levels. So Mary is my director of studies, and this is kind of the wider scientific group. So as mentioned, they've worked on a wide range of different species and different questions in evolutionary ecology, and Andrew came in as a sort of beekeeping supervisor, opening us to the world of access to lots of beekeepers who were doing lots of conservation and things like that. So it's funded by NERC, my PhD, but we've got some additional funding from the Eva Crane Trust, which I'll speak about a little bit later on. So right, the first thing I wanted to do, because my PhD is quite genetic based, I do a lot of different, there's a few different methods, and there's a couple of ideas that I wanted to speak about before I get into the sort of data side, just so that we're all on the same page. Obviously, we've got a mixed audience here today of beekeepers and scientists. So I wanted to talk about genetic markers briefly. So it seems like a scary thing, but actually a genetic marker is just a genetic sequence that's used to observe variation either between species, individuals, populations, but it's just a sequence of DNA. And there's two sort of broad sources of DNA in our bodies and in honeybee bodies. So you get nuclear DNA, which comes from the nucleus, and it's all wrapped up in your chromosomes, and that's nuclear DNA. So the important thing about this is that nuclear DNA is what codes for all of our characteristics and our traits. So anything from like a biochemical process all the way up to your physical phenotype, so your characteristics. And then you've got these organelles inside cells called mitochondria, and you hear this a lot, but they're the like powerhouse of the cell, and they have their own little genome as well. But it doesn't code for sort of your individual traits. That all come from your nuclear DNA. And I'm going to talk about two different types of genetic markers in my talks. So I know we might have seen a bit of this in Dylan's talk, but I just briefly wanted to mention it. So you've got microsatellites, which are these repeated regions. So you can see CTA is a little motif that's repeated here in this individual 5 times, 6 times, 7 times, and they're quite changeable, so they're quite good for looking at demographic processes. And then we've got SNPs. The long word of that is singular nucleotide polymorphisms, but essentially that's just a base change, so an individual base change. And these different markers kind of roughly fall into 2 kind of categories as well. So you get neutral markers, so they're non-coding. So an example of these is microsatellites and mitochondrial DNA. They don't code for anything that would change your characteristic phenotype. But they are good for telling us about demographic changes and gene flow, and they are really useful. And then you've got the other type of marker would be a functional or an adaptive marker. And these are in coding regions. So those are the things that code for these functional processes or characteristics. And an example of them could be SNPs, or indels is another one. I'm not going to talk about those, but it's just to give you a rough idea that there's other markers out there, and they can be both neutral and adaptive. So a SNP can be inside a coding region, but it can also equally be outside a coding region. And basically different methods and different markers are used to answer different questions. So it depends what you're asking and as to which one you'd use. So with that technical bit out of the way, what I'd like to talk about is the first study that I did on my PhD. This is the first chapter, and it was changes in genetic introgression in Apis mellifera mellifera in Cornwall. And I'd just like to give a shout out to Gabrielle, because a lot of this data was generated by her, and it has, I believe some of this data was actually published in a paper by John Ellis and her. So, so these Cornish populations that we're talking about here isn't generally Cornwall. What I'm actually talking about is the B4 group. So Andrew mentioned that he's the director of this group, and what they are is a conservation group down in Cornwall that they breed, and they want to increase the range of AMM. And they had used microsatellites to monitor their conservation program. And that got sent away to Apigenetics, and it's microsats. We've just looked at this, but just to remind you quickly what I'm talking about. So they would send these samples away. And what I did was look at 2 data sets. So we've got a data set from 2015 and we've got a data set from 2018 that are bees in this conservation program. So I looked at 2 data sets and 2 different analysis methods. So I've got structure and encore. So I'm going to look at 2 data sets over 2 different analysis. So the first thing we're going to look at is a structure analysis of the 2015 samples. What you're about to see is a table, and this table has standard populations in that are used to kind of calibrate what groups we're about to see, but and they'll have numbers in them, and the numbers are probability, they're Q numbers, they're called membership values to different groups. So what you can see here is the group. So group 1, group 2, and group 3. And then you've got different populations this way. So you can see Italy, Austria, Austria-Hungary, Slovenia, Sweden, France, Norway, and they are standard samples of different subspecies. So looking at them, I can tell what group that the program has created is representing these different subspecies. So I can see that my standards for AMM have jumped into group 1, and the AMC standards, so these Austria samples here, and there's another Austria one down here, have jumped into group 2, and the Italian one has come out on its own near enough in this group 3. And then you can see at the bottom here is Cornwall 2015, and it's got some membership values there. So we can see that 0.71 probability here is grouping with AMM, 0.28 AMC, 0.02 AML. So this is Carnica and Ligustica, obviously. And we can visualize that in plots. And I know people have seen some of these earlier in the week, but this is a structure plot. So each of these colors, you can see there's 3 colors here, are representing these groups. And the numbers along the bottom are just representing these individual populations. So you can see that AMM, group 1, is represented by this red grouping here. AMC, group 2, is represented by this green color here. And then AML, which is the Italian population, the Ligustica, is represented in blue here. And you can label those up so it's a bit easier for you to look at. And on the end here is Cornwall 2015. So you can see where it said here 0.71 is because the majority of that is this red signal. And then you've got 2.8 of it, which is this green signal. And right at the bottom there, you can see some Italian grouping there. Each line is an individual bee, and what we're doing here is visualizing the whole population. So you can see like there's one coming up the middle there, that's an individual bee that has a higher proportion of AMC than the others. So then we can look at 2018. Don't worry that we're looking at these separately. I'm going to look at them all together at the end in a summary slide, but it's just to give you a look through the data. So the same thing's happened again. We've got our groupings. So we've got everything has changed around. Unfortunately, structure doesn't allow me to keep the same colors in the same groups because you run it a number of times. But so group 1 here is AMM now. Group 2 here is AML. So this is the Italian one. If you can go and find the Italian populations just here, and AMC. So the Carnica populations jumped into this cluster here. And at the top is Cornwall 2018. So 0.82 with AMM, 0.02 with Ligustica, and 0.15 with Carnica. And again, we can visualize this in a plot. So again, the colors have changed as well, just to make it extra confusing, but I can go through it. So group 1, AMM, is now green. So AMM is green, AML is red, and AMC is blue. That's group 3. And on the front here, this is Cornwall 2018. So you can see there's a predominant green here. And again, each line is an individual bee, but we're looking at that as a whole population. So then I did something called an encore analysis. Now, encore works slightly differently. So what happens is I create the baseline populations. So I take all of the Carnica standards and I turned them into a baseline, which is just a group. I've pre-grouped them together. Whereas in structure, it was grouping it itself based on an algorithm. I'm doing it and then I'm checking what the Cornish population looks like against those baselines. So the actual description of it is that the samples are assigned the highest probability of belonging to a baseline population via the probability that a given genotype would be produced by that baseline population. So this is the 2015, they call it a mixture analysis. So these have been created. So I took all, like I said, I took all the standards and created these baselines. And then this is 2015. And then I say, well, what mix of Cornwall, what mix of the Cornish 2015 population would spread across these? What's the probability? So you get 0.64 for the 2015 with AMM, 0.36 with Carnica, and nothing for Liguistica. And then I did the same thing again for 2018. So we've got 0.86 for Mellifera, 0.14 for Carnica, and nothing for Liguistica. So that's all well and good, but let's have a look at what it all looks like together. So the structure analysis, what we see from 2015 to 2018 is an increase in AMM. Liguistica or AML, it stayed the same. So we didn't see any change there. And Carnica, we see a reduction. And we can do the same for Encore. And we get an increase in Mellifera. Liguistica stayed the same, and Carnica, we see a reduction. So you're seeing the same pattern across both. But what I would say is that the amount of change is a difficult thing to say. But even though the amount of change is a difficult thing to say, I would also treat the results with caution, because it's a very small sample size for this experiment, because I was just matching up things that happened to have been sampled over the 2 years. So it wasn't, I didn't purposely go out and sample these, it was what happened to be in those data at the time. But it's a bit, it's a small sample size, and you've only got kind of 2 points, 2015 to 2018. So it's a bit like having 2 points on a graph, and it's not necessarily a trend. And what would be better would be to do them more repeatedly, and possibly with the SNP chip, it's going to be more accurate. Okay, so that was the first study I wanted to share with everyone. So we know about the conservation group, B4, but what we wanted to see is there anything in the wider Southwest population that might be of interest to, weren't necessarily part of the B4 group, but were doing conservation. Sorry about that. Okay, so what do we examine in this experiment? So again, I contacted, a bit like Dylan did, I contacted beekeepers who thought they had native or near native bees and didn't import. And in this experiment, we looked at SNPs, so different to microstats. And this work was done along with some colleagues up at the Roslin Institute. So Mark Barnett and David Bragg are working at the Roslin Institute, and have done some good work on honeybees. And we said, hey, why don't we do some work together? So the SNP chip was run with these guys, and we used this SNP panel that was designed by Alice Pinto's group. So these SNPs have been evaluated as being really accurate in assessing integration in AMM bees from C lineage bees. So when I say C lineage, I mean Carnica, AMC, and AML, Liguistica, are grouped together in that C lineage. So how did we exactly go about this? I just wanted to orientate people as to where we are when I say the Southwest, because I know this is international, there's a lot of people from elsewhere. So we're talking about this area here. The University of Plymouth is right on the edge of that, it's about there. And we, again, targeted those native, near-native beekeepers, and we got about 98 samples. This group here was a group of beekeepers who were really interested in what we were doing, and they hadn't had their bees genotyped before, so it was quite good and sort of spreads by word of mouth, and took as many samples as I could. Some of these dots represent more than one colony, because some people sent more from one apiary. So this analysis, this experiment is performed on an individual worker bee. And their DNA was extracted, and interestingly, their DNA was extracted from a group at the Rosling Institute. So they have a citizen science week there. This is Mark Barnett, who's got a little map up, and I think he's talking about bees, and here they're teaching people about genetic code, basically. And so they did the DNA extraction, and then I was sent the raw data, and I used a custom Python code to format that, and then it was filtered for missingness in Plink to make sure we've got enough SNPs across all the samples. And in the end, 80 of the SNPs passed in all of the samples. So I know I said I had 98 or whatever it was, but there's a lot of standards on that as well, which come on the end of the SNP chip when you get the data. So that data was then processed in a program called Admixture, which is kind of similar to Structure, and it was visualized in R. Okay, so this is a similar thing to what you saw on the structure plots. Each line is an individual bee, and what you notice probably straight away is that we've only got two colors, right? Red and blue. And that's because K = 2, because the C lineage ones are grouped together in the SNP chip. So you're looking at Carnica and Ligustiga integration into AMM. So you get a blue signature here. These are obviously each line's an individual bee, but this is predominantly blue for Carnica, predominantly blue for Ligustiga. And these are some these are the standards actually that come with the SNP chip when you get the raw data. So yeah, you've got a red signature here from the AMM standards, and then this is the larger Southwest population. So what you notice is you've got some quite high and you've got some quite low. It's quite a mix, but there's a few that were quite good. So let's have a look at that in numbers so you can sort of see. So the mean Q value, so this is that membership probability value for all of the samples being AMM, is 0.72, so 0.72 probability of being clustered with AMM. And then C lineage here. And there's a fair bit of movement in this data, and that would be because there was a fair bit of range in the different values that we got. So the highest was 0.94 and the lowest was 0.2. So this is not it's a very low, it's a high C lineage value and a low M lineage value. I've got another data set that sits alongside this I'm not going to talk about today, but it's a RAD-seq experiment. So for the scientists in the room, I guess you might know what RAD-seq is, but it's like sampling the genome using restriction enzymes. So you're subsetting the genome really, or subsampling the genome. But yeah, I've got pooled and individual data that sits alongside this. But the conclusions from the SNP chip run was that a lot of the Cornish populations are integrest, individuals are integrest, and it's not shocking given the history of importation that we have in the UK. Okay, so I guess the next logical question there was like, what's the wider picture for, so we've looked at a conservation group, we've looked at the wider Southwest, but then there's real questions that remain about what the picture for Apis mellifera and mellifera is in the wider population of the British Isles. So this was some extra funding that we got from Eva Crane. I had a few things that didn't work out, which I'll speak about in a bit. So we went for some extra funding to do some more work. But what we want to know is, we know about islands, we know about Colonsay, we know about the odd sample in the Southwest that's pretty good. But we wanted to have a look elsewhere. So we asked beekeepers from across the British Isles who suspected they had native or near native bees to submit samples. So same thing again, but in a much larger geographical scale. And this time we used pool whole genome sequencing. So when I say pool, I mean a pool of colony, a colony pool. And I guess like, why do that? Well, a single honeybee is not representative of a colony due to the multiple mating of the queen. This is a similar thing that Matthew was doing and mentioned in his talk, where they did like I think it was like 500 bees they put into their analysis for doing some QTL experiments and things like that. And it's a similar thing to what Grace is doing over in Ireland. But basically honeybees are quite a complicated biological system. And in order to capture that kind of colony level integration and colony level information, this is the way that we've gone about it. There's pros and cons, and it's a difficult data set to work with, but I think it's worth it to answer the biological question at hand. So, and whole genome, because there's a couple of things I'm asking about where integration is happening across the genome. And there's actually an additional experiment within this data set that I'm currently working on, which I'll mention at the end. Okay, so let's have a look at the sample sites across British Isles. So you can see we've got a pretty good spread. And we took representative samples from, so we've got some from Scotland, Wales and Ireland, and we know there's groups working there, and they will be doing a much more in-depth look at those places. What I've done is sample sort of the select ones from there. And then the rest of them, I don't think have been sampled before on this kind of scale. So it's quite exciting, it's quite interesting. So we did pooled colony whole genome. It was 30 bees per colony, and we've got 75 samples from across the British Isles. And then we had these standards from people across Europe. So I want to say thank you to everyone who sent me bees, especially the guys who I just contact out the blue, who've never met me before and then send me bees. So it's really helpful, and it's nice to work in a field that's so collaborative. There's also another data set alongside this, and it has some standards in it. So I've kept those in the data set, and I will explain what this is all about in a moment. But in total, with all of the standards, we've got 177 samples. You might also notice that I've got a Buckfast sample in with this. Basically, I've left that in at the moment, but I'll explain why. So methods, what did I actually do? So at the University of Plymouth, I dissected 30 bees per colony. I weighed their thoraxes, and they were extracted in batches of 5. So they're donating the same amount of tissue. They were then extracted in batches of 5 and then equimolarly pooled. So they donate the same amount of DNA. This sounds like something that I just mentioned really casually, but originally this experiment had 189 colonies in it. So, and I did 30 bees from every colony. So I dissected over 5,000 bees. It took months. It takes a really long time to do it this way, to make sure everything's donating the same amount of DNA and tissue, but there's reasons that we do that. But then it was sequenced on a BGI platform, which stands for Beijing Genomic Institute. So it's sequenced over in Hong Kong at a depth of 50. And then we get into this, you get your raw data, and there's a million quality steps. So I filter all the breeds, make sure they're of good quality. If they align to more than one place, they're thrown out. If they don't have a pair, they're thrown out. And then you go through this next pipeline, and by the time I get here, I think I had just under 12 million SNPs. But a lot of those will be erroneous. They're not true SNPs. So what you need to do is keep quality filtering, and then I do some of my own filters on the end, and eventually you get 9 million high confidence SNPs. So after all that, I'm going to show you some preliminary data now. So I'm still kind of working on this, but let's have a look at the admixture graph. So this then was placed into Plink like the last experiment, and then after being in Plink, it was filtered for missingness and all this stuff, but it eventually goes into admixture as just 2 files. So this is just preliminary because I've left the Buckfast in, because what I was doing was just having a real early look at the data. So I placed everything in, but ideally I would take this out. But so we've got this, this is Carnica, Ligustica and Buckfast. It's not surprising that Buckfast is quite C lineage grouping, this big yellow signature here, this yellow grouping, because a lot of people suspect that, you know, Buckfast is mainly made up of C lineage bees. And then Apis mellifera and mellifera standards that get this red signature. And then this is the British Isles, and it has its own sort of this blue signature here because K equals 3, so we've got 3 groups, 3 colours on the graph. Yeah, so I've got a blue signature here that is predominantly coming from the British Isles. Okay, and we can look at the numbers that pertain to that graph as well. So again, it's K equals 3, so there's your 3 groups. And you can see that Ligustica, Carnica and Buckfast are grouping in group 3, and this is the British Isles, 0.29 probability of being in here. Here you've got Mellifera, 0.83, and the British Isles is giving a 0.51 probability for this one, average basically. And it's got its own, it's the largest member of group 1, which is that blue signature. Okay, so as I said, ideally I'd like to take Buckfast out, but I just did a really quick look at the data, and then the high performance computer unfortunately went down at Plymouth. It's only just come back online, so I couldn't quite get that done for the conference, so sorry about that. But yeah, so C and M. So if we think about this in terms of locations and thresholds and sort of see what that really means, I'd like to put it into sort of these broad categories. So anything above 0.95. So there were 3 samples out of the 77 samples that made it through filtering. And there are 2 in Ireland and 1 is in Colonsay. So that's not really surprising. We kind of knew that already a little bit from other studies. And I've placed those on the map. I don't know if you can see them. There's 1 and then there's 2 here. And then there's another 3 that if you bring that down to 0.90, you've got 6 in total, so including these. And those are Inverness, and there's an island one, and there's 1 on the Isle of Man here. The Isle of Man one's quite interesting because I think they have really strict rules about taking bees on and off of the Isle of Man because they don't have Varroa. And then if you bring that down even further, obviously a few more jump in, and we've got Newcastle, Limerick and County Kildare jump in there. That colour is slightly close to the background of the map. So if I do that, you might be able to see those 3 popping up. So there's 1 there, 1 there, and 1 there. I don't know if you can see that. And then if I bring it down even further, there's just 1 more that pops in, and that's Jersey. So it's quite interesting preliminary findings. Like I said, there's more work to do with this data. The other thing I can do is you can thin it out basically. So remove some SNPs. It's almost like too much noise in the data. So the thing I want to sort of take away from this, I'd like to discuss a little bit further from this study is this idea of thresholds, because it's a question that comes up quite a lot. You know, when is AMM still AMM? And beekeepers are the ones saying, well, what level of integration is acceptable for me to breed from? You know, when does its trait start to break down? When is it AMM anymore? And to answer that question was a big thing that as a group we wanted to answer, and it's something Mary was quite keen on. And you sort of got this, to answer this question, you need phenotypic information, so traits information about bees, and adaptive markers, which we spoke about earlier. So preferably SNPs that are all the way across the genome. So when I joined my PhD in 2017, we were like, right, we're going to set up a survey. So in 2018, which was the first summer of my PhD, I wrote a quite large phenotypic survey of UK bees, and we were going to get all this different trait information. I was going to do all the genetics on the bees as well. And this is everything I asked everyone to measure. So I can probably feel beekeepers in the audience now cringing a bit as I asked them to do all this. And this is where it comes in that I'm not a beekeeper. So I arguably asked way too much, because out of 152 people who joined the survey at the beginning, 2 finished. So that wasn't going to work. That wasn't going to be enough data for me to answer that question. And so not to be defeated, we tried again. And in 2019, we set up another phenotypic survey. And I asked for less information, less often. And the participation and finishing numbers were really improved, but they were still far from what would be useful. So out of 124, and 11 people managed to finish it. And it was at this point where I was like, I don't know how I'm going to answer this question. And I got an email from a beekeeper saying they'd heard about a research group in Sweden. So I popped them an email. And that's when I got in contact with Sonia from the University of Skövde. And we started having a sort of back and forth about what each group was doing. And we've actually discovered it would be really good collaboration. So they've got a project that's called Supporting Nordic Brown Bees. And they have a team in Sweden. based in 2 locations, and they have a team in Norway. And I'm not going to go fully into what they're doing because they've got a talk on January the 19th, and it would be really, really great if people could make that. And they'll go into exactly what they're doing. And I think they recently published a paper. So the main idea behind this collaboration was that the general gist is that they're doing phenotypic measurements of colonies. So this is them at an apiary in Sweden, and it looks lovely. And we were like, oh, well, we can genotype your colonies. So this is us on a bridge after doing some bumblebee sampling in Dartmoor. But so they've got this phenotypic information, and we have genotyped all their colonies. So that's the extra experiment that's tied up in the UK-wide data with all those buckfast bees and all the other standards that they've got. And hopefully pairing that genomic information with phenotypic monitoring is going to allow us to identify regions of the Apis mellifera genome that's associated with traits, and we can start unpicking this idea of, when is AMM anymore, and where is that integration happening with the other data set. So it's going to be quite an interesting data set, and I'm really looking forward to it. Thank you very much for listening. That was kind of a blitz through. It was a bit quick, actually. Sorry about that. But I just want to say really a massive thank you to my supervisors who are constantly helping me and giving me feedback. And thanks to the funders for paying. And then thanks to all the beekeepers. The beekeepers of the UK are super helpful. When I started my PhD, it was really daunting because I don't know anything about beekeeping as such. And Beecraft magazine put out adverts, as did Bibber, and it's been really amazing to work with everyone. And thanks to the collaborators, we work in I think a sector of science that's really great for everyone kind of mucking in together and sharing ideas. And SciCam is a really good example of that. So thank you.
Vic, thanks very much for sharing with us all all of your latest results, but I think it nicely highlights, hang on, let me just push this back. I think it nicely highlights the different scales that you've been working at, from the very local right up to the UK wide, and now obviously working with partners in Sweden and Norway, which has been a fantastic extension to the project. I guess one of the things for me, I think at the moment we don't have any questions yet, but I think you highlighted in your talk that, one of the important questions that's come up repeatedly, it came up in Dylan's interesting talk a few weeks ago, and also when Matthew was talking about, what we're trying to achieve, I suppose. And I think it's quite a central question for us to discuss is, on the one hand, we've got conservation of Apis mellifera, the dark honeybee, across, its European range. And we were talking to Gabrielle earlier about, there still seems to be people that are questioning what that native range actually is or was to start with. But on the other hand, the optimization of locally adapted traits and ecotypes. And those two things are related, obviously, but not necessarily the same thing. So I guess I was just generally interested in your thoughts on that, and perhaps anyone else's thoughts on that, really.
Yeah, it's obviously something we've spoken about a lot in our research group, but it's sort of these slightly different but very similar overlapping ideas, and it can be difficult to sort of tease those apart. So like the idea of conserving AMM is arguably slightly different from the idea of bee improvement, of having that as a local ecotype that you then use to improve on. Yeah. And this, I know John has mentioned quite often about the idea of like land races and having that as a locally adaptive form that you then pick and improve. And they are slightly different, intertwined. They kind of overlap in your mind, and it can be really hard to not let that happen. But it is, what are you, if you're trying to conserve traits, are you trying to conserve AMM? Are you trying to, it's a difficult thing to know, and if everyone's on the same page, especially with, so I guess I guess it's an interesting thing to talk about, and it's a very difficult thing to pick apart for some people.
Yeah, it would be really interesting at some point, I think, to have a wider discussion with people in the group about that, I think. Anyway, sorry, I've sort of sidetracked you because there's a number of other questions come in. So Grace says, nice talk, Victoria, well done. Interesting you picked up hybridization in Galway. Connemara are struggling with recently introduced bees after trying to keep AMM isolated for a long time. It looks like you showed the difficulty with imports. So I guess that was a comment from Grace, but
yeah. So generally, the island samples did come out quite high. I would also take these results with a pinch of salt, because those numbers change depending on what samples you involve, and I would like to thin that data out. So that is a really preliminary look. They're not going to change drastically from something being, 0.2 up to 0.8, but they will change slightly. And when you're looking at those thresholds, that's what matters, isn't it? If it changes slightly and it jumps into another threshold, well, what's that threshold and what do we choose? But yeah, I will be sending, just in case there's any beekeepers in the audience who are yelling, where are my results? I've literally just gotten this data, and I will be sending out results to beekeepers who sent samples as soon as I can.
Okay, great.
To individuals, yeah.
So Clive also has a kind of relevant to some of the other points we were just mentioning, is an interesting point about, we hear increasingly about the alleged detrimental effect of managed honeybees on populations of wild bee species. So if AMM is considered a UK or European native species, how does its presence square with the argument?
Could you say that again? Sorry, beginning.
So I think the question is around, we've got this, this conflict, I guess, between people on one hand arguing that these are completely managed, this is a completely managed system, and that they're actually detrimental to native pollinators.
Oh, I see what you're saying.
On the other hand, we've got, these bees are actually native to the UK and to a lot of Northern Europe. So how do we square those two arguments?
I would first say that it's more of an idea that it would make sense that honeybees at a high density might put pressure on native, bumblebees and things that aren't being looked after by humans. But the evidence for that is still kind of scant. I don't think there's enough of a robust body of literature that's done studies about that. And that's what I would say needs to be done first before we start making decisions on where you can and where you can't, and how many you can and how many you can't, because we don't know yet. I don't think there's that's what I do. I don't think there's a solid foundation to say.
Yeah, and also ties in, doesn't it, to Grace's work in Ireland on feral populations, and obviously some of you will be aware that there's been some recent reports of potential feral populations in the UK that are potentially native, and how you square those with managed populations. Yeah. So Grace then asked, do you think the pool-seek approach is picking up hybridization that genotyping of a single bee is missing? So for example, if you're using microsatellites or SNPs of single workers, that you're going to be missing that kind of level of detail in your data?
You absolutely could, couldn't you? I think if you were going to use the SNP chip, I would recommend that you do a couple of bees from a colony and see what you get, see what that spread looks like. So there's pros and cons to using pool-seek. I'm not sure that pool-seek at the moment it's not really going to be available to mass people, because it's an incredibly difficult and technical thing to do. But yeah, you could be. The other thing is that admixture and a lot of programs are built for individuals. So when I've put a pool in there, what I've done is I've created a composite genome from that colony. So it's like a pseudo individual, okay? Because it won't take anything that's not biallelic sites, so it has to be 2. So that's all you can put into it. So it could have a confounding effect on it, because that individual might not really exist. I don't know. And also you lose linkage. There's a lot of things with pool-seek that there's pros and cons to using both methods. But I think if you're using pool-seek, ultimately you are capturing a genotype of the entire colony. Also depends on how many bees you use. There's a lot of difficult things to be said about it. It's a really difficult data set to work with. And I think you are capturing more information about the colony. Absolutely. Yeah, there's no argument there.
Yeah.
Yeah, I think
balance
it's a trade-off between doing the two things. Ultimately, Pool-seek's not going to be available for beekeepers at the moment. So doing an individual bee is still better. Like if you can't do anything, do a couple of bees from your colony. When we're talking about doing large studies about QTL or GWAS, then Pool-seek is absolutely the way to go. And because there's been a couple of studies using individual bees, they don't get any associate do because you can get these allele frequencies. I can dig out allele frequencies of the whole colony because I know how much DNA has gone in from each bee. So I get balanced allele frequencies and things like that. So yeah, you are getting more information obviously from the colony.
Okay. Matthew Richardson's asked, the preliminary data for the UK is very interesting. Were there any trends in progression across the UK regions or in populated versus remote areas?
Yeah, I haven't gotten that deep into that data set yet, so I apologise, but I can't really answer that yet. I sort of have looked at where was the ones with the least integration, and that's what you saw in that map that I clicked through. But no, I haven't looked at that yet, I'm afraid. Sorry.
Okay. Well, yeah, hopefully once you get a chance to work up those data better, you'll have some further answer on that. And Norman asks, thank you for your presentation, Vic. You covered the cormos. Did you sample any wild colonies? Oh, sorry, no, that's the wrong one. Sorry.
That's okay. In the answer to that one, I haven't sampled any wild colonies. So.
Okay, when we come back to that. In the study that involved British Isles genotypes in blue, so in your structure plot where you had the blue signal, are you saying that these are uniquely British Isles genotypes? Which I think is a really interesting question, actually. Are they AMM or are they other foreign genotypes that you didn't have standards for?
Good question. So obviously the majority of that signal came from the British Isles. And if you think about it, those standards are from across Europe, and the British Isles bees, if they're not imported and people have been keeping them for a long time, maybe they've got their own, background genetic signature. It doesn't mean that they're their own subspecies. It just means that they have their own background signature, which they will because they've been breeding with each other. And these are specifically people who try not to import, So yeah, obviously there's always room for more standards in these data sets because that's how these programs work. But at the moment, I would think it was a background British signature, basically, of genotypes. Yeah.
Okay. And then, sorry, you've already partly answered this, but I think one of the interesting things that Vincent asked about, if there is there is signs of movements or of a movement that people are interested in encouraging kind of ferial colonies and so on. Have you got any impression, or I guess any of the beekeepers here, is there any opinions or views on whether that's something that we should be encouraging or not? And I guess that's one for you, Vic, but perhaps for anyone else who wants to contribute.
Yeah, well, it's difficult because I'm not a beekeeper, I suppose.
No, I know.
Yeah, sorry.
But have you had any impression from beekeepers, for the beekeepers you know about, whether that's something that they would, welcome or not?
Inherently, the beekeepers I speak to are people who, if I think I understand what you're getting at, they're people who don't import and want to keep near native bees anyway. They are the people I try to contact, and I contact them through groups like B4, within BIBA, and actually even within the BBKA. I do feel like they're moving away from that idea of imports when I spoke to one of the heads there, but difficult to say. I think maybe Andrew might be better at answering that question. He speaks to beekeepers all the time.
Okay. Sorry, I'm kind of going through these questions just as they appear, but sometimes we're sort of jumping back and forth, but I can't really avoid that easily. So John Mosdale says, you seem to have very limited data from Wales, a western part of the UK where AMM might be expected to be commonish, but you don't seem to have contacted the Welsh Beekeepers Association. Was that intentional or an oversight?
No, not at all. So the way I contacted them, because there's people working in that area, so Dylan, who spoke earlier in the week, we have a small group that we kind of speak to. So the guys at the Roslin Institute in Scotland, Grace in Ireland, and the Bangor group. So actually we had a meeting and I contacted people through them a lot of the time, and it was people who'd already been involved. So I didn't purposely leave anyone out, if that's what you mean. I also was only taking a few representative samples from those places, as Dylan did. I think part of his PhD was to look at the Welsh population. So that would have been something he was doing, I suppose. But no, I didn't intentionally not contact the Welsh Beekeeping Association. I actually gave a talk there in my first or second year, getting participants. I did. So I've given a talk to the Welsh Beekeeping Association on one of their, I can't remember when that was, 2018, about the survey and about they would.
Yeah, but as you say, I think it was also because we were working collaboratively with Dylan.
Yeah, and I didn't want to sort of sample all of his bees and usurp Dylan. That's, we're trying to work together. So
yeah. So Sonia, hi Sonia, nice to meet you remotely, I guess. Great talk, Victoria. Thank you so much. Looking forward to the ongoing collaborations. Do you have any idea why the British beekeepers were not really interested in taking part in your survey?
I think the first one was too onerous, and I think that was my fault. I don't think that was any. There were a lot of people who signed up, and everyone was very keen. Actually, I think that I designed the first one badly, which happens. PhD is a learning curve. And then the second one, I'm not so sure. So I'd given them pictures to estimate different things from, so they could just give me a rough estimation of like coverage on their frames and things like that of different things. And I was asking them to do that every month. So that was kind of as scattered as I could go with my data, to not be as onerous on them. But then I think because it was once a month, maybe it just slips through the cracks. They've got other things to do. I do think there's an overlap of beekeepers who want to get involved, beekeepers who are also very busy. So
yeah, true. Joe Widdicombe, who's hi Joe. One of the beekeepers in the southwest that's been very helpful in providing samples and was doing a fantastic job in keeping AMM populations here. But he says, I think from a beekeeper's point of view, we can approach it in two ways. And I think Joe's referring to the initial question about conservation of AMM and locally adapted forms. One is to do with conservation when purity of a population is important. And the other is from a bee improvement point of view, where the important thing becomes breeding true. So hybridization makes it difficult, but perhaps absolute purity is less important. And yeah, I think from my perspective, that's right. But I think what would be interesting to have a collective discussion about is what we don't want to do, I suppose, is one to end up trying sort of negating or in conflict with the other. And I think having both is important there's a conservation perspective, and also important from a bee breeder's perspective about they have bees that they have desirable traits that they want to work with.
I guess if I can.
Yeah, sure, please do, Gabrielle. Yeah.
I guess it all depends on how much pure stock you still have available. When there is very few and the majority of bees are kind of introgressed, you rather work with those and decrease a little bit your threshold.
Yeah.
But when you have a good population with a lot of still pure bees with a high threshold, you might consider culling out the others before you start breeding and improving.
Yeah,
you have to weigh those factors for every population individually.
Yeah, absolutely. And I suppose the other thing that we're not sure about, of course, is that whether some of the AMM we're picking up could also have been the result of some imports. Of course, we don't know that those are necessarily bees that originated in the British Isles. And again, that's another interesting research question that we might try to tackle.
Has Norman done a paper on that together with the installation of the Colonsay Reserve?
Well, I think it seems reasonably well. It seems like a complete no-brainer to me that, of course, we would have originally had native AMM. I think the question is just whether all of the AMM, currently we're finding in the UK are direct descendants of that original native population, or whether there's some, there might be some of those that have already been imported from continental Europe, for example, as AMM.
Is that a killing factor, like a kill factor for their conservation? I mean, No,
I don't think it is, but I think it's that interesting. It's that interesting interface, isn't it, between local adaptation of forms, for example, that are particularly adapted to Cornwall, for example, which may have been there as a remnant populations for thousands of years, or may not have been. We don't know.
Yes. Yeah, true.
So it's quite a complex story, really.
I see. Well, I have another question, sorry, before
Go ahead.
I go on. I was wondering about, with the SNP analysis you showed, there was this mellifera group, and that seemed to be very introgressed. So I was wondering what kind of reference population that was.
So those, when the guys from the Roslin Institute send me the data, they send you these standards on the end of it. So they weren't samples that I went and sampled. They were ones that came with the data from the Roslin Institute. So pass. I presume that I think you'll find, actually, I did look them up in Alice Pinto's paper, and actually they come with the SNP chip. They were used originally to build that SNP chip, and I think that's when they started partitioning things off and making sure they could get as close to the whole genome results. So I think they actually come along with that. For you to run, because you can't run this admixture analysis without standards in it. Yeah, you would have to.
I'm familiar with the reference sample set from Alice, but there wasn't that much introgression in there. Like, it was like almost no.
Yeah. Okay. So pass on that. So what the guys at the Roslin ran it through their pipeline, and I ran it through my pipeline, and we crossed them over together, and they came out the same. So like the Q values came out the same for my samples as it did with theirs. So we'd have to go back and ask them. But that, I mean, the analysis comes from the raw data set as well. So whatever bees have been run through that, I still get their original calls, their base pair calls, and then that's just turned into another file and put into the program. So I guess that would be with the Roslin Institute, unless they put their own standards on the end. That's also a possibility. But yeah, so I got the same integration levels. Pass. But I can get back to you on that, actually. Unless,
because usually you're using reference sets with like purer stock and not fully introgressed. So it seemed like there was no one individual that was like predominantly pure AMM.
Yeah. Okay. Yeah.
So, I'll have to check. Okay, we'll ask Mark. Okay. Thanks.
So Grace again had just a comment really, but I think it's relevant to that discussion about what this signal is telling us. But we've found the same signal, Irish bees, standing out from other EU samples using microsatellites. So similar, this blue signal that Norman was asking about. But we're slow to publish that due to the issue around reference samples, the same one. But maybe this signal is real and added evidence for protection of local biodiversity. So again, coming back to those themes of what are we actually looking at? But hopefully we're now starting to get the kind of data that we can start answering those questions.
So and then we're looking forward for like follow-up talk.
Yes.
Where you clear up all this.
Where I've cleaned up the data, it's not preliminary anymore. Yeah, okay. I'll be back.
And then Joe commented that after 150 years of importing, we may simply never get back to a pure population. And I guess again that kind of refers back to this conversation about what are we aiming for? And I think that's true, that we may well never get back to an entirely pure population. But certainly there are areas and pockets in the UK, Vic. I think you would agree that. So. Ivan then asks, thanks Victoria for sharing interesting facts and figures. Incidentally, I'm interested in the BIBA initiative. Do you think you could elaborate further regarding backgrounds and interests from the BIBA group regarding native bees?
Yeah, so actually originally BIBA were it says it there, but they're the Bee Improvement and Bee Breeding Association. And I think there was a small group of them that were more interested in AMM bees. Andrew would know more about this because is it Nick Bentham Green? Is he still involved, Andrew?
So essentially the B4 group really came out of BIBA, and the crossover is even deeper than that, in that the original BIBA members were set up SciCam. So I'm not quite sure, everybody's crossing over with everybody, and that's fantastic. Does that answer the question?
So I think it was more about what their sort of interests are, and I think that says it all, is because I think I think B4, like Andrew just said, they came out of it. And I know a couple of BIBA members, and they're generally interested in AMM, but I think they had originally were a group about bee breeding. They've got a really good website and contact details on there if anyone is interested in it.
Okay, thanks. So Sigron says, thanks very much for your interesting talk. Great, it's great that you deal with genetics. Therefore, I have a question that's not directly related to your talk. Beekeepers sometimes observe a phenomenon that their honeybees mix, so a mix of Carnica and others, and they become darker over the years when they're not treated against Varroa, which is interesting. These observations come from Germany, where we actually don't have AMM anymore. How could this be explained? Thank you for your answer.
So when they're not treated for Varroa, they're becoming darker. So Varroa is obviously quite a strong selection pressure on bees. I don't know why they're getting darker per se, but what you have to remember is, like, we put a lot of pressure on our bees, and there's a difference between what a bee wants and what a beekeeper wants sometimes. And I think I don't know why they're getting darker, but that would be a strong selection pressure on those bees to survive Varroa, basically. So over the years, if you've got the same colony breeding, then maybe they're shifting towards something else. But I couldn't tell you why they're getting darker, unfortunately.
Interesting. I don't know if any. I mean, yeah, I don't.
Any beekeepers?
Yeah, if any beekeepers have observed that. I mean, obviously, it's a really interesting observation, and another kind of caution against us just using one. And I know that nobody here does this, but using one aspect of their phenotype, such as color, as a discriminatory feature for, you know, AMM compared to the other subspecies. Yeah. Vincent says, Victoria, your results show that the dynamic of the global honeybee population is increasing. I'm just checking that's correct. Has your project got any societal implications within the organisations you work with?
Does it have any implications for the individual societies I work with? I think once it's peer reviewed and I write a paper, it would be a really good reason to tell people to stop importing. I think even if you weren't into AMM bees, you're not particularly interested in dark bee per se, but you just want your bees to survive and to be good bees and to disease not be coming into the country and have this continued genetic influx breeding with your bees that you're trying, even if you're just trying to improve your bees and the person next door is importing bees from, I don't know, the south of Europe every other year, they're not adapting to the climate and to your area. And I think that is the main implication of kind of work that we do. I don't think any scientist, beekeeper or not, or bee scientist or not, would recommend continually importing something that can just breed with the local population freely. They're not going to have a chance to adapt, basically.
I suppose the other, the other encouraging thing is just that the breeding programs that people are setting up seem to be the initial signs are that these are effective when they're increasing the. Is that right, Vic? Yeah. Yeah,
I would say so. I think especially with the B4 group, they do have a massive advantage of being surrounded by water because they're based in Cornwall. And I know, for example, Joe Widdicombe, who asked a question earlier, who has some quite pure AMM down on the Vrain Peninsula, is the only beekeeper there and is surrounded by water completely because he's on a peninsula. So, you know, those things obviously really help, but they also practice things like drone flooding and all kinds of different methods. So I think it helps to get together with a group of like-minded beekeepers and start doing your thing, because one beekeeper alone is, you know, if you're surrounded by people who aren't wanting to do what you do, it's kind of a futile exercise. So yeah, don't know what I'm saying anymore, but I recommend you all get together in big groups and start working together.
Yeah. Norman, actually, yeah, this is really interesting, Norman, because I think it goes back to the conversation we had a minute ago about So Norman says there are some beekeepers who are convinced that AMM in parts of the UK are purely descendants from Dutch or French AMM. If they are Dutch, French or Dutch AMM descendants, it therefore invalidates conservation efforts being made in the British Isles and Ireland. And Norman says, he disagrees. And I would disagree as well, but I think it's, again, this question of, there are two kind of separate things, aren't there? There's the question about AMM, and then there's a question about locally adapted traits that are really useful for bee breeders. And they're not mutually exclusive at all, but they're two slightly separate questions. So I don't know if you want to make any further comment on that, Vic or John, whether you wanted to make anyone else wanted to make any comment on it. It's just an interesting.
It might be worth mentioning that in one of the comments earlier, Grace made a comment that perhaps there'd been evolution of local ecotypes to Ireland and the UK since the historical colonization, and that would be interesting to get more data to investigate that question, I think.
Yeah, I think we did say, I did read that one out about Grace's microsatellite data, suggesting that there might be these particular signatures.
Yeah, and she'd also commented about ecotypes underneath another one of the questions as well.
Okay.
Yeah.
Anyone else? Vic, do you want to have any final comment on that? You don't need to.
No, I don't. Not really. I kind of agree with that. Even if bees even if AMM bees in the UK came from old imports from wherever, Holland or somewhere, it doesn't invalidate the fact that they should be protected.
No.
But we don't know that, and you'd need like museum samples, or I don't know what else you would do that with. But so we don't know, but it doesn't invalidate the fact that they should be looked after.
It's always a question. From when do you start accepting the adaptation process? I mean, they don't. Yeah. That's the question, isn't it? I mean, this study from Collis has shown that the local adapted bees, the local bees do better than the imported bees. They haven't been necessarily the native bees.
Yes.
So also there were bees imported, and after a while they were adapted, and it didn't even take them too long to adapt.
Yeah.
So I think this question is kind of futile. If there are black bees, even if a portion of them have been imported, there's always migration among the populations. So, it's kind of, trying to get arguments like desperately against dark bees.
Yeah, no.
Sounds like it. Why?
It does seem sometimes like, they get quite a hard time, I think, in the UK. It seems it's quite, I don't know what it's like actually in other countries, but it does seem like sometimes people are very, very, very reluctant to accept that these are, long-standing native species. And I'm not quite sure where it all comes from, really. The kind of anti-honeybee.
It's the same. It's not only in the UK. It's the same everywhere. It's kind of the syndrome. The grass is greener on the other side of the fence. So it kind of started that way. So that's where the import started. We notice here when every time something goes wrong, major or minor, it's always the genetics. Like you can claim it on the genetics, and you're quick to get other bees from somewhere else before you look at your own, the way you treat or manage your bees. You'll question that only in the end. First, it's the genetics to blame. It's kind of a very known syndrome.
I just want to point out there's one more question appeared in the bottom of the chat, maybe as the final question.
Oh, sorry, I missed that. Thanks, John.
It's from Barbara Keaton, and the question is, in certain places in mainland Europe, there are conservation areas where imported bees and transhumans are not allowed. I know we have some certain remote Reedsdale or island populations, but is the UK too small to attempt this?
Well, we have it even in Switzerland, and we're much smaller. So no, I would say no from my heart. UK is not too small to attempt this.
No, I think there have been attempts to have some bee reserves. I think, Andrew, didn't you have a bee reserve in Cornwall? There's an area.
Yeah, we've made several attempts to do things, and it's difficult.
Yeah.
Getting everybody to agree.
Yeah.
It's kind of disappointing because, you know, if they just stopped importing stuff, then I think we have big agro businesses, a very small number of people who gain financially, and you know, there's maybe only 200 individuals out of many tens of thousands who are involved in this stuff, and you know, the finance is just driving it.
Yeah, I mean, Maggie's just posted a, you know, a relevant further comment, really, that imports are popular surely because many beekeepers are looking not so much for survivability but for profit from honey. I mean, certainly some of the big commercial beekeepers will be, but as you say, Andrew, they're relatively limited in number, aren't they, in the UK, the big commercial importers?
The people that are importing the colonies are a very small number. I mean, there's maybe 200 of them in total, including individuals. You know, so it's big business. It's big money. You know, and it's, you know, if you can buy a queen for 15 quid, which means that you don't have to inspect your colony, that saves labor costs. So you just, you know, you accept the fact that each year or each other year you change the bees, and it's a very European thing. You know, 1 in 10 of every single queen in Europe is requeened with a commercially produced queen. I mean, that's mainly from the south of Europe. So this is not a, you know, we're only at 8.5% or so requeening from foreign queens. You know, Europe's ahead of us, and the States are requeening, you know, some of their commercial operations are requeening twice a year. So they don't mind if 30 or 40% of their hives die off because they're just sticking in another queen because it's, you know, cheap. They're not that bothered what happens to the populations around them or the beekeepers around them, who form the majority of the beekeeping population. And on a brilliant side, there are 1 or 2 beekeepers who are working native bees and using less intensive input, and producing really good outputs of honey. You know.
Well, I guess ultimately that's the thing that's going to change things if you can demonstrate that they can do that, I suppose. That would be the most convincing argument.
Actually, it's not like the honey is not only the, you know, the major driving force. There will be a talk from Switzerland, from Padrot Fried, about our attempts to install more than this 1.5 kind of reserve we have. And that has failed despite having loads of money from governmental parties and so on. So it wasn't a question of money or anything. So there is other forces working against reserves. This will be very interesting. Kind of depressing, but, you know, good to learn from.
Yeah. There's just a couple of other comments that are worth sharing before we sign up. So Vincent said a couple of things about, and it was coming back to this point earlier, and it relates to a lot of Grace's work about feral populations and the potential importance of feral populations for, you know, effectively providing a gene pool for mating. But I guess one of the issues is, you know, is it feasible, Vic, do you think, for large feral populations to be existing still within the UK? Or
so there was someone who did a PhD on it at Leeds, looking for feral bee colonies and trying to track them. I think over 2 years or something, and most of them weren't there for very long. I think Ireland is slightly different landscape to a lot of England, especially because there's not that many old woodlands left, I would think, in England, and that's where they would sort of reside, I suppose. So she didn't find any that persisted for a very long time in that PhD. And obviously, like, it's really good to have that background feral population, and it's exactly what you would need to leave those kind of, the idea of this local land race that's just left to do its thing. The issue with that is that obviously honeybees breed with their local population. And if someone's still importing bees and you've got a feral or wild population living in a tree, and it's still a real issue, this issue of importation. And I think that's what we need to solve first, in my opinion. That's the real thorn in the side of anything moving forward.
Okay, thanks, Vic. And then finally, Sigrun was asking again about this issue about a self-sustaining population. So in your opinion, how large does a protected area for AMM need to be in order to have a self-sustaining population?
I would be guessing if I gave you a number to that, if I'm honest. So I don't know. Obviously, there probably needs to be some studies on how far it's going to travel outside of that reserve area. Obviously, a lot of people have, like, I know the one in Switzerland is in this special, you know, in between some mountains and things like that. We obviously can't really do that in the UK.
It's like a huge valley, but it's open to the plain. So it's very hard to manage. It needs a lot of money and effort. Already there. Yeah.
Maybe it's maybe worth commenting that there are some general rules of thumb that people apply in conservation, like 5,500 or 5,000 rules about how large populations need to be to avoid inbreeding, and then the negative effects of inbreeding, which is inbreeding depression. There's a lot of controversy in the literature about whether those rules of thumb can be generally applied, and that you need specific data about the demographic structure of the particular species that you're dealing with, and so on and so forth. So it's hard to come up with a general rule of thumb, and I guess it takes some more thought to think about the specifics of honeybees, the fact that they're haplodiploid, that the queens are polyandrous, what the natural nest density would be in the area, and think about those kinds of things to get a rough answer. But it's difficult to state and think.
Yeah, and I think those kind of rules of thumb that are kind of brandished around in textbooks are actually quite unhelpful, actually, because so much will depend on the individual system and the particular characteristics of that. I don't really think we can apply particular rules, but you're right, Joan. I think that you need to know a lot about the ecology and demographics of the population as well as the genetics before you can start assessing that. But I mean, I guess at some level, you know, we have to start somewhere, and I think any area is better than none, and the bigger that area that can be, the better. Because of course it's not just about geographic area, it's about the density of bees, of the population within that.
Yeah, you need to, I mean, even if it's a small area, when there is enough variability to start with, you can go for a while, and then you can always do some, you know, migration between reserves. Yeah. Maybe within the UK, so not to stir up this discussion again. I think, yeah, I agree.
Yeah, and I guess, you know, things like you know, Collinsay and so on are nice examples of how, at least in the short term, you can set up very successful pockets, if you like, of these populations.
Yes. Rather have a small but manageable population. I think the isolation, that's more the killing point for reserves. You know, when they're not isolated enough, you cannot manage it. You cannot keep the purity, you know, at a wanted level.
Yeah. So just on that, Grace has added that because Grace is very involved in this, you know, studying feral populations in Ireland. She's saying she's hearing loads of cases of wild honeybees in the UK, and her PhD student Catherine's PhD was quite small. Oh, sorry, Catherine was the PhD that you were talking about. It was quite small in size, and she matched wild bees with nearby managed bees, which could have been then biasing our sample area and so on. Okay. There are lots of old oak forests in the UK and actually all over the UK. Oh, I'm missing it. So interesting work to come out of the UK. Norman has a PhD student working on this too now. I didn't know about that, but that sounds interesting, Norman. So fingers crossed they're adapting and surviving there too. Yeah. So that seems like a positive and nice note on which to end on. But I just wanted to obviously say many thanks, Vic, for taking the time to share your interesting data with us. And obviously there's more interesting things still to come out of those data sets. Thank you very much. And I also wanted to obviously thank everyone else for attending and for such an interesting range of questions that we've had. And to make sure, I guess, sorry, the one other thing I wanted to just acknowledge was that we forgot to mention that one other, as well as Andrew, John and myself supervising this project, Vanessa Hummel was also supervising this project, who's been really important in terms of some of the bioinformatics side. Vanessa's not here tonight, but we wanted to make sure that we acknowledge it. So, I just wanted to sign off really by making sure everybody's aware that we've got a double bill apparently coming up next week from Dr. Olga Pruns from the National Institute of Agricultural Sciences in the Republic of Korea. And I think that's a slightly more complicated format. Perhaps, Gabrielle, I think you were maybe going to give us some more detail of that.
Yes, so as Olga is like 7 hours away from us, she has sent us her talks and we will present them to you, and you will have the opportunity, the ones that participate live, to ask your questions, and we would then take the questions and record them, like have the discussion at another time point with Olga, and you will be able to see the Q&A session after streamed on the conference platform, as also her talks from then on. So
and I think somebody
had an opportunity, this online format, also to reach colleagues in South Korea. I find this
it's great,
thrilling. Yep.
And Gabrielle, somebody had also asked, I think, just before we go, somebody had asked whether the chat questions are recorded, because for those people who are not, English isn't their first language, it's maybe sometimes difficult to follow them all just in live time. So yes. I don't know how that works in the recording.
Well, we cannot keep all the questions, in written form because it gets kind of wild, but then the Q&A session is recorded and also translated in the subtitles, but you have to give us a few days, be patient a little bit, and then you can go and watch it later on. So all the questions are in there.
Okay, great. Well, thank you very much, everyone, and look forward to seeing many of you next week for August talk.
Yes, thank you very much to you all. Thank you. Bye.